Greetings to everyone. Welcome back once again to this educational channel on biology. Today we'll be discussing the Pahang Bio Paper 2, Year 2021 paper based on the KSSM syllabus, the new syllabus, and this is the third set of questions. So the questions discussed will be questions 7, 8, and 9. Questions 7 and 8 are structured questions, and question 9 is an essay question, right? So we'll be revising some biological concepts and also learning the techniques of answering the questions. And also there will be a very easy memory tip for you to help you remember certain facts, right? So let us increase our pace of learning from snail's pace and zoom in straight into our lesson. The first question here is based on Chapter 14 of the Form 4 syllabus, Support and Movement in Humans and Animals. Now, diagram 7.1 shows the human muscles involved in bending the arm, right? Then, in diagram 7.1, name tissue P and Q. Okay, so we have to write the names here in the space provided. So what is P? Now, P connects muscle X to bone Z, right? So this tissue must be a tendon, right? And Q connects bones together, all right? And helps to keep the bones in place at the joint there, right? Prevent them from being dislocated. So Q is ligament, all right? Connecting the bones together. Next, explain the difference in characteristics between P and Q, between the tendon and ligament, right? So we still refer to these two tissues as P and Q, all right? Don't use the words tendon and ligament, as you will not be answering the question directly if you do it, right? Now, both these tissues, P and Q, are strong. The tendons and ligaments are strong. But there's one very big difference, and that is in their elasticity. Okay? So which one is elastic and which one is not elastic? Right? So P is strong or P is not elastic while Q is elastic. If you write the word strong, there's no marks for it because it's not a difference. It's a similarity. Right? So P is not elastic while Q is elastic. Right? So if you can't remember, you have to reason it out. Now P is not elastic because it has to transmit the pulling force of this biceps when it contracts to the bone. Now if it is elastic like a rubber band, when the muscle contracts, when the biceps contracts, the tendon will just stretch, but it cannot pull the bone, which is the radius here. So if it's elastic, it cannot carry out its function well. So it's not elastic like a thread, like a strong thread, okay, or a few pieces of these threads, all right, so that it can transmit the pulling force of the muscle to the bone, okay? It can help to pull the bone upwards. But Q is elastic, the ligament is elastic to allow some movement of the bones at the joint here. If it is not elastic, then it will restrict the movement of the bones, such as the radius and ulna. Okay? So that's the difference. P is not elastic, Q is elastic. Now next, if tissue P is torn because of an accident, what problems will be faced by this patient? Right? So if P is torn. Now this is another way of asking you the function of the tendon, but in a negative way. All right? So if P is torn, that means it cannot carry out its function. And what is its function? To transmit the pulling force of muscle X to the bone, right? So if it is torn, it cannot carry out this function. Then state the consequence after that. So let's look at the points. First point, tissue X, which is the biceps, will not be attached to Z, the radius. If it's completely torn, if the tendon is completely torn, then tissue X, the biceps, will not be connected to Z, the radius. Okay. Pulling force from biceps cannot be transmitted, or from X or biceps cannot be transmitted to Z, which is the radius, through P. Okay, so we refer to the tendon as P, as in the question here. All right. Now, as a result, the movement at this joint here, the hinge joint here, will not occur, will not happen. All right. That means the arm cannot be bent, cannot move upwards, because the tendon will not be able to pull the bone anymore as it is torn. Okay, and when it's torn, it's painful, so the person cannot pull the bone up, right? So this is 2 marks, so any 2 points. All right, but it's better to write in all the points to make it a complete answer. And also, in case if one point is wrong, you still have another point to back it up. Okay. Many students are confused about the tendon and ligament, right? Which one is elastic, which one is not elastic. So to save yourself some thinking and reasoning, let's see what we can do here, right? So this is a very simple and easy BioKey Memory 8. Tendon, T-E-N-D-O-N. The T-E, and the E stands for elastic, but there's a T here, so we can use the Malay term for no, tidak elastic. Of course, if it's in Malay, there's a K there, right? It's in BM, there's a K in place of C, but I just use the English word. One Malay word, one English word. Tidak elastic. T-E for tidak elastic. Likewise, for ligament, we have an M-E here, M before E. M-E can mean misty elastic. Okay? Or just remember for tendon, the T is tidak, so tidak elastic, that means ligament must be elastic. Next question, B. Based on diagram 7.1, explain how bending of forearm takes place, which involves muscle X, Y, tissue P, and bone Z, right? So we use these letters when we explain about the bending of forearm. So underline this part. It's the bending of forearm, not the straightening of the forearm. Right, so first you have to identify all these tissues that are mentioned. X is the biceps, Y is the triceps, P is a tendon, and bone Z, which is the upper bone, is the radius, right? The lower bone is called the ulna. The ulna. So we are talking about radius here, Z. So mention all of this in the answer. Now, the first point is muscle X contracts while muscle Y relaxes. Okay, so biceps here will contract, whereas triceps, which is muscle Y, relaxes. This is the action of antagonistic muscles, which work in opposite ways to one another. Okay, so when one contracts, the other will relax. So in this case, for the bending of forearm, it is muscle X, the biceps, that contracts, right? Then when it contracts, there's a pulling force, but it has to be transmitted to P, the tendon, all right? Because when it contracts, it will shorten, so it will pull P upwards, and P will pull the bone Z upwards also. So the pulling force of X, the biceps, as it contracts, is transmitted to, first, to transmit to Z, it must go through P. So the pulling force of X is transmitted to Z, the radius here, through P, the tendon, right? To pull the bone upwards. So causing Z to be pulled upwards, causing the radius to be pulled upwards, and then the ulna will also follow. And that is the bending of the forearm. Right, so notice that all the tissues are mentioned, X, Y, P, and Z. And don't mention the name. Okay, you must have, you want to write the name, you write it in brackets for all the different tissues. But the letters which represent the tissues have to be mentioned, okay, according to the question. Next, bone Z is affected by a type of disease shown in diagram 7.2, right? Then, in your opinion, suggest a food that is suitable for this patient. So if you look at this diagram here, you have to identify the disease shown. As you can see, a lot of holes, okay? black holes here in the tissue, right? So this shows that there's loss of calcium from the bones. And the name for this disease is osteoporosis, which is loss of bone mass caused by loss of calcium from bones, right? So in a normal bone, it will be more, there'll be more bone tissue and there'll be less of or smaller holes, not so big, okay? So osteoporosis literally means the porous bone, all right? Osteo is bone, porosis means porous, okay? Porous bones which have a lot of spaces in between them, empty spaces like this, right? And this disease is caused by loss of bone mass, caused by loss of calcium, right, from the bones. So you're asked to suggest a food. So here, suggest a specific food. Don't just write dairy products, okay? So our answer can be milk, cheese also can, right? Green vegetables, anchovies. In Malay, we call that ikan bilis, right? Anchovies, anchovies, or even a vegetable like broccoli. So all these contain high calcium content, right? And it increases bone strength or mass or bone density, right? So since this is 2 marks, we should not just mention the food, we should mention why we choose the food, and why we consider it suitable for people who have osteoporosis. So P is 1 mark, and any of these explanations, E, is 1 more mark. It's better to write down all 3 points, which comes together like this, all right? Right. The next question here is based on Form 4, Chapter 11, Immunity in Humans. Diagram 8.1 shows the action of an antibody and antigen in an immune response. Now, based on diagram 8.1, explain the interaction between the antigen of the bacterium and the antibody. Okay? So, bacterium is singular form. It's a singular form. Bacteria, plural form. Now, let's look at this picture and analyze it. Here we see the Y-shaped antibodies. All right? Normally, they are drawn as Y-shaped, and they are linked, okay, or bound to the antigens here. So this antigen is the surface particle, okay, found on the bacterial cell. It's part of the bacterial cell, and it's a foreign substance that is recognized by the body as something foreign. So these antigens are foreign substances that stimulate the production of antibodies by lymphocytes in order to destroy these antigens that have entered the body. Okay, so what we see here is that the antibodies have bound to the antigens to form the antibody antigen complex, where they are linked together like this. So the specific antibody here binds to the antigen of the bacterium at the antigen receptor site of the antibody, which we cannot see clearly here, but in the textbook it is mentioned in page 203, okay, the Form 4 textbook. The antigen molecule will combine or bind with the antigen receptor site found in the antibody molecule. Okay, so remember, the antigen is the bad guy. It means antibody generator, antigen. And the good guy is the antibody that's trying to destroy these antigens, which may cause disease in the body, right? So the specific antibody binds to the antigen of the bacterium at the antigen receptor site of the antibody, and this forms an antibody antigen complex, as we can see here. So whole thing is called the antibody antigen complex. Okay? And as a result of this, agglutination occurs, right? So agglutination is the process whereby the bacterial cells are clumped together. They are linked together. All the bacterial cells are linked together to form a big clump so that they can't move around, and so that another cell called the phagocyte can come and engulf and destroy them. All right? So these are the 2 points. That's the interaction between the antigen and the antibody. Okay? Now, agglutination is only one method of action of the antibodies, right? Think of the word glue, agglutination. So we know that it causes the bacterial cells to stick to each other, to stick to the antibodies, and then they are linked to each other to form a clump, right? Now, other than agglutination, the other ways or processes whereby the antibody can act is through lysis, opsonization, precipitation, okay, and neutralization, which are not shown here. Okay, so you have to recognize this is agglutination, okay, because you see all the bacterial cells, the 3 bacterial cells here clumped together. Next question, explain how the binding of the antigen and antibody will overcome the bacterium attack. Meaning to say, after this binding process occurs, what happens next? All right, to destroy the bacteria. Okay, so the antibody causes clumping of the bacterial cells. Okay, when it binds to the antigen, the antibody will cause all the bacterial cells to clump together. Okay, now this is the common term used in most biology books. Textbook use the word coagulates the bacterial cells. So I suppose if you write coagulates the bacterial cells, it's accepted because it's in the textbook. But the common term used is clumping, so write both down. Clumping of bacterial cells coagulates the bacterial cells, and the process is called agglutination, as seen here. Next, to destroy them, okay, this makes them easy targets to be trapped and destroyed by phagocytes. Okay, so imagine after these clumps of bacteria are formed, the phagocyte will move in and extend its pseudopodia to engulf this antibody-antigen complex, and then it will form the vacuole, right? And after that, it will secrete enzymes to digest these bacterial cells and kill them. Okay, that's the job of the phagocyte. So the phagocytes will work together with the lymphocytes. Lymphocytes produce the antibody, the antibodies, but the phagocytes engulf and destroy the clumps of bacteria. Question B. Diagram 8.2 shows the graph of changes in the antibody levels in the blood of individual A for a specific period of time. So this graph shows you the concentration of antibodies against time after the first injection and second injection of a substance. So what is the substance? Looking at the shape of the graph, it's like two hills. One is lower, the first is lower, and the second one is higher. So this shape tells us that the injection is the injection involves injection of vaccine. Okay, not anti-serum. It's a vaccine. And vaccine, what is a vaccine? It is a suspension of weak or dead pathogens that is injected into the body of a person to stimulate his or her lymphocytes, right, to produce antibodies. Antibodies to destroy the antigens or pathogens in the vaccine. Okay, so that's why the level of antibodies increases after the injection, because the lymphocytes start to produce antibodies to destroy the antigens or the pathogens in the vaccine. Okay, now based on the diagram, diagram 8.2, describe the differences in the response to the first and then second injection. Okay, so the response to the first injection is here, starting from maybe around month 1. Okay, increases, the level of antibodies increases and then decreases down to low level. Then in the 4th month, the second injection is given. So this is the second response, or response to the second injection. Okay, this one, the curve here, right? So let's look at the differences and compare and state the differences. Okay, here I show the graph from my video because you can see here the gradient more clearly. So let's look at the differences. So this graph is similar to the graph here in the question. Okay, now in the first injection, antibodies were produced slowly over a few weeks. Okay, you can see here. So how do you know that the production of antibodies is slow? Because the gradient is low. Gradient, the value of the gradient is small, gradient of the curve. But in the second injection, after the second injection, antibodies were produced more quickly. You can see the curve here. For the curve here, the gradient is steeper and is bigger, higher value. Okay, so this shows that in the second injection, the antibodies were produced more quickly than the first injection, because in the second injection, the lymphocytes have already been exposed to the pathogen already, once, okay, during the first injection. So they are able to produce antibodies more quickly, okay, because they have already been exposed to the antigens earlier, all right, in the first injection. Then after the first injection, this is the second difference, antibodies were produced at a low concentration, okay, the maximum level was around here for the first injection, but it has not even reached the level of immunity. But in the second injection, the greater production of antibodies occurred, leading to a higher concentration of antibodies. So the peak here is higher, right, for second injection than for the first. Okay, here we are talking about the amount of antibodies produced. Okay, it's different. The first one is about the rate at which the antibodies are produced, for the first difference. Now the last difference is, after the first injection, level of immunity was not yet achieved, but after the second injection, level of immunity was achieved at the 6th month. So looking back at our curve here, after the first injection, level of immunity was not achieved because the level of antibodies never reached the level of immunity. This is just, it's a maximum here at this level. Maximum level of antibody achieved was only at this level, much lower than the level of immunity. But after the second injection, the level of immunity was achieved at around 6 months, where the 6th month, when the level of immunity, the line intersects with that of the curve. Okay, so the person becomes immune at this stage, okay, around the 6th month, because his level of antibodies have reached the level of immunity. All right, so any 2 differences out of the 3 to get 2 marks. Question 2 is a hot question. The process of creating a vaccine against viruses and bacteria takes several steps. Okay, it may involve several steps. The step begins with the genetic codes of the virus, which are determined. So in your opinion, why is this step so important? Why is it important and must be taken in producing a vaccine against a specific disease? Why do we need to know the genetic code of the virus? All right, so let's look at the answer here. The reason is to take out specific parts of the viral genetic code to weaken the virus, okay, especially the parts that make the virus like dangerous and infectious. So when we take out certain parts of the viral genetic code, the virus becomes inactive, and then the virus is still alive but weakened already, unable to cause infection. And then this is going to be put in the vaccine, so that the body, when a person is injected with this vaccine, which contains the weakened virus, the body will be able to recognize the viral genetic code and respond by producing specific antibodies to destroy the virus. But the virus will not be able to attack the body and cause the person to fall sick. So that is an important aspect of the vaccine. It must not be able to overcome the immune system of a person, right? It's just to provide a sample to the body of the person that's vaccinated, a sample of the virus, so that the lymphocytes of the person can be stimulated to produce specific antibodies to destroy that particular virus. Okay? And also to prepare the body for future, in case there's future infection by the same virus. Then the body will be prepared. And it will have memory cells or lymphocytes that can produce the same type of antibodies, specific antibodies against that particular virus, which it has been exposed to, which the body has been exposed to after the vaccination. Here's a hot question from me for soalan ramalan, based on the COVID-19 virus. Okay, so this is my question, not part of the question in the Pahang paper. All right, this is my own question. So it may be important because we are nowadays, many people are injected with the vaccines for COVID-19, especially the mRNA vaccine. Let's find out more about the mRNA vaccine. So here's a statement. Some vaccines against COVID-19 contain strands of messenger RNA or mRNA that has the code or blueprint for production of the spike protein of SARS-CoV-2 virus, right? Then By applying your biological knowledge of vaccines, explain how this mRNA vaccine works to produce immunity in humans against the COVID-19 virus. 4 marks, or it could be more. So the special thing about this type of mRNA vaccine is that it does not contain any parts of the virus. It does not contain the dead or weakened virus at all. Neither does it contain the spike protein. Okay? It contains the strands of mRNA that has the genetic code for production of that spike protein from the virus. Now, in this first picture of the coronavirus here, right, you can see the spikes coming out from its surface. This is the spike protein, and it looks like a crown like that. That's why it's called coronavirus. Corona is crown, means crown. So this spike protein, all right, the scientists have sort of investigated the genetic material of the virus, okay, and so they are able to come up, okay, with the mRNA that has the code for the production of this spike protein. All right, they know the genetic code of the virus already. So it is a strand of RNA, messenger RNA, that has the instructions to make the spike protein. Okay, so this is the substance that is put into the mRNA vaccine, which is injected into the body of a patient. Okay, so the question is, how does this mRNA vaccine works? Okay, let's read from here. After the mRNA is injected into a person, cells in the person's body will use the mRNA genetic code to produce the spike protein of the COVID-19 virus. Now you can see here, once the mRNA is injected into a person's body, it will go into the normal human cells. It will go into the human cell, and the human cell will then use the genetic code in the mRNA to produce the spike protein. It's sort of like a process that just automatically occurs because there's a mRNA inside there with the instructions to make this protein, right? So the spike protein will be produced on the human cell. Okay, it's produced by the human cell, not the virus, all right? And then the immune system, which are the lymphocytes, will recognize that this protein or antigen is foreign. Now, although the cell is a human cell, the antigen here on it is the spike protein. It is a foreign substance. Okay, so this is recognized by the lymphocytes, which will then be stimulated to produce specific antibodies to destroy the spike protein. So these are the antibodies, right? So in this way, the antibody against the COVID-19 virus is produced, and also memory cells are produced. Memory cells among the lymphocytes are produced, which will later be able to produce the same antibodies, the same specific antibodies, in case there's an actual infection of the COVID-19 virus in the person. Okay, so memory cells antibodies against the virus are produced. If the individual is later infected by the actual COVID-19 virus, the lymphocytes will quickly produce enough antibodies, enough of these specific antibodies, and immediately destroy the virus before it can cause serious illness. Okay, so these antibodies were produced because of the spike protein that was produced by the human cell. All right, so this is how the mRNA vaccine works. That means only the first step is different. The rest of the steps are the same. The first step, when the mRNA is injected into the person and causes the person's own cells to produce the spike protein. That's different compared to other vaccines. Let's go on to section B, which are the essay questions. Now, the total marks for each essay question is 20 marks, okay? And you only answer one question out of the 2 questions in section B, all right? So make sure that you answer all the parts for the question. Like 9A, B, C, must answer all. Don't miss out anyone. So let's look at this question 9. Diagram 9.1 shows a food chain in a paddy field, all right? Grass eaten by a grasshopper, which is eaten by frog, and the frog is eaten by the snake. First question, state the definition of food chain, right? So food chain is a sequence of energy transfer from one trophic level to the next trophic level, starting with the producer, for example the grass, and ending with the secondary consumer or tertiary consumer. In this case, it's the tertiary consumer, the snake. So the producer is eaten by the primary consumer, and primary consumer is eaten by the secondary consumer. Secondary consumer, the frog, is eaten by the snake, right? So, and the food chain is a sequence of energy transfer, all right? Because when, for example, the grasshopper eats the grass, energy is transferred to it in the form of the food, which is then, when it eats the food is digested to form nutrients like glucose, which is absorbed by the body of the grasshopper. And then the glucose is broken down in cellular respiration to produce energy, right? So the energy from the grass has been transferred to the grasshopper, right? And then to the frog, and then finally to the snake, right? So it's a sequence of energy transfer from one trophic level. So grass is at the first trophic level. Trophic here means food, the first food level. Okay, first trophic level, followed by the grasshopper, second trophic level, frog, third trophic level, and the snake is at the fourth trophic level. Okay, so 2 marks, one for each definition, with the keywords: energy transfer, trophic level, producer, tertiary consumer. Let's look at question B now of the essay question. Diagram 9.2 shows a method to estimate population size of snails in a farm by marking on its shell. All right, so marking on the shells of the snails, the symbol X or something. Now, explain the technique. All right, so we have to recall this technique that's used to estimate population size of animals. And the name of the technique, the short form, is Cmrr. Cmrr. So the method used to estimate the population size of snails is capture, mark, release, recapture technique. C for capture, M is mark, first R release, and then second R recapture. Okay, remember Cmrr technique to remember this full name. So what is the technique that is used here? First, you have to capture the snails. So snails are captured randomly in the farm, because it's in the farm here. And let's say it is 10 snails. You capture 10 snails. All the snails are marked with an X on their shells, like what's shown in the picture, using non-toxic paint, Indian ink, all right, or any type of mark paint that is non-toxic and more permanent, so it won't be washed away. And also, it must not make the snail obvious to the predators. Okay, then all the marked snails are released back into the same area so that they can mix randomly with their friends. Right? Then after a week, about 7 days, or after 5 days, not after 2 days, maybe 2 days, the snails, they move so slowly they haven't mixed with their friends yet, right? Not so you have to make sure that they have mixed with their friends. So after a week, a few days to a week, maybe 4 days, 5 days to a week, the snails were captured randomly a second time from the same area. Also, we must not just capture the snails that are marked. Just a random thing. That means that you must just capture any snails that you see. Don't care whether they are marked or not. So this is the second capture. Then after you have recaptured them, you have to count. The total number of snails in the second capture is referred to as Q, and the number of marked snails in that total number of snails. Okay, you must count how many snails are marked. Let's say you captured 20 snails. So count how many of the snails are marked. Okay, maybe 5 of the snails are marked. Okay, so record these two numbers, okay, in your table. So the estimated population size of the snails is calculated by using the formula. Population size equals to P times Q over R, where P stands for the number of snails captured in the first capture, which were all marked. And then Q is the total number of snails in the second capture, and R is the number of marked snails in the second capture. Okay, so how do we get this equation or this formula? Okay, let's try to understand how it works. So I'm going to explain to you how we derive the equation. you see, actually in your first capture you marked all the snails, right? So we can say that there's a, out of S, S is the total population size of the snails, P were marked. So P over S forms a fraction, which is the same as the sample that you took after a few days. So when you recapture the snails, or you capture the snails again in the second capture, this is your sample, where your total number of snails is Q, but out of that R was marked. So P over S equals to R over Q. It's the same proportion. All right. The sampling method is, the results of the sample is R over Q. But P over S is the ratio or the proportion of snails that were marked out of the total population. So the proportion of snails that were marked out of the total population equals to the proportion of snails that were marked out of the total number of snails in the second capture. Okay, it should be more or less the same. So when you have this equation, then your S you bring over this side of the equation, right? And the R and Q you bring over this side. So it becomes Q over R. So S equals to P times Q over R. That's where you get this formula here. P times Q over R is the population size of the snails. Okay? So in case you forget the formula, you can still derive the equation, derive the formula from this equation. All right? Okay, so we need to memorize the steps of the Cmrr technique. Next, let's look at this question. The graph in 9.3 shows the prey-predator interaction. All right? So the prey is the rabbit here. You can see the dotted line, right? Rabbit is eaten by the fox. A fox line is the one that is smooth line, not broken, the one below here. Usually the predator will be in smaller numbers, right, compared to the prey. If you are given two graphs and not told which one is the prey, which one is the predator, you can more or less identify the prey as the organism with the higher numbers or bigger population size. Okay, usually the prey is small, but the population size is bigger than that of the predator, usually. All right? Okay, so describe the interaction between the prey and predator, meaning the rabbit and the fox. Okay, so the first point is that the rabbit is the prey that's eaten by the fox. So the fox is the predator. Right? Now, describe the interaction. So you have to explain. Let's say I want to explain this part. Okay, somewhere around the year, this part, 1925. Now you can see the prey population increases very high until a very high amount, and then is followed by the increase in the predator's population. Okay, so how to explain that? Now, let's read from here. When the population of the prey, that means the rabbit, increases in 1925 here, long time ago, right, the peak here, the population of predator also increases after a short while. You can see here, the peak also comes. All right? Because the predators, the foxes, will have more food, more rabbits to eat, since it has such a high population of, the field may have a high population of the rabbits, right? And so when the predators have more food to eat, it will result in a higher rate of reproduction for the predators. So predators will have more babies and be able to reproduce and produce more of the offspring. And they'll increase the population size of the predator here, okay, the fox. However, when the population of the predator increases, around this part, maybe around 1929, the population of the prey starts to decrease. I can see here it goes down. Okay, so common sense will tell us that as the predator's population increases, it will eat more prey, so the number of prey will decrease. Because more prey is eaten by the predator. So you see the dotted line going down here. After the peak for the predator, the peak for the population of predators. Okay? Then, in this way, the prey population is brought back down, okay, after having such a high or large population size. The population size decreases and goes down to its minimum amount, right? But then when it goes down to the minimum amount here, now the predator population size will decrease because it will have less food ready to eat. Then when the predator's population size decreases, the prey population begins to increase again. So this will start the cycle back again. Okay. So when the population of the prey increases, this will be followed by the increase in population of predator. And then when the population of the predator increases, this will be followed by a decrease in the population of the prey. Okay, and that will also cause the population of the predator to go down when it doesn't have enough food. So the prey-predator relationship overall helps control the populations of prey and predator to be at dynamic equilibrium. So you look at the population of the rabbit, it increases, decreases, increases, decreases, but there is an average value around this part, okay, over which the population of the prey fluctuates or increase and decrease. So that's called dynamic equilibrium, where the population of prey and also for the predator fluctuates, increase and decrease around a fixed average value. Okay, due to the prey-predator relationship, the prey and the predator, they both affect each other and affect the population size. So this helps to maintain the balance of nature. So dynamic equilibrium means a stage where the population of the prey and predator fluctuates or increases and decreases around a fixed average value, like this red line here for the prey. Now for the predator, the curve is lower, so the average value should be somewhere around here. Let's now look at the last part of the question, which is question D. Diagram 9.4 shows 3 recycle bins. These 3 bins are used to collect used material, and that is used paper, used plastic, glass, and used plastic. Justify how the practice in recycling of the used materials in diagram 9.4 can contribute in maintaining the balance of nature. Okay, meaning we have to explain how this practice helps to preserve the environment, protect the environment and the animals and plants. Okay, right. So, and also prevent the damage of the environment. So we have to divide our answer into 3 parts, because these 3 materials, paper, glass, and plastic, they are produced differently. For example, paper is produced from trees, right? So if we recycle the used paper, we will help to prevent the trees from being chopped down, because we can reuse the paper again. And less trees will be chopped down. Okay, so when less trees are chopped down, there are many benefits, and it's the opposite of deforestation. Okay, so your answer is that just writing the opposite of the effects of deforestation. So these are the good effects. When less trees are chopped down, there are more habitats for flora and fauna. Okay, so these habitats are preserved. For example, the tropical rainforest will be preserved because the trees in there will not be chopped down so much. All right, and habitat is the place where the organism lives. So when the habitats are preserved, it means that less or no food chains or webs are disrupted or disturbed. They just carry on. The food chain will be as normal. And then the extinction of flora and fauna is prevented, right? Because this flora and fauna means the animals and the plants will have a habitat to stay in. So they will not die of hunger or die of loss of the habitat. All right. So when the extinction of flora and fauna is prevented, this will preserve biodiversity, meaning the many different types of animals, plants, and organisms on our earth. It will preserve the biodiversity. If one of the animals were to die, that means the biodiversity will be decreased. But if we preserve the flora and fauna, then there's no extinction of these organisms, then the biodiversity is preserved. Now, next, talk about the air. See, the first part is talking about in the forest, what happens. So now what about the air surrounding the forest or in the forest, the air above. Now, concentration of carbon dioxide and oxygen in the atmosphere will be maintained. Please spell out in full, CO2 and O2, carbon dioxide and oxygen, is maintained, and this will lessen the increase in carbon dioxide concentration in the atmosphere. So nowadays, the carbon dioxide concentration keeps increasing in the atmosphere because of the burning of fossil fuels. But if we preserve the forest, the trees in the forest will absorb the carbon dioxide for photosynthesis, right? So they will decrease the concentration of carbon dioxide in the air. And they will release oxygen through photosynthesis too, which is good for organisms to use for respiration. Okay, so trees can help to lessen the increase in carbon dioxide concentration in the air, okay, by absorbing the carbon dioxide from the air for photosynthesis. Now, water catchment area, the forest is preserved because we didn't cut down so many forests, cut down the trees in the forest. So water catchment area is an area that receives the rainwater and then collects it for a while. So the forest is a water catchment area, and it is preserved because we didn't cut it down. And then this will prevent flash flood, soil erosion, mud flood, landslide. Okay, so all this is on the land, when on the hill slopes. If the forest is not preserved, then trees will die there, and then the roots cannot bind the soil together. Then when it rains, there will be soil erosion and landslide. But in this case, if we preserve the forest, the trees bind the roots, the trees roots bind the soil together. So the soil will not be eroded, and then they will not flow down the hill and cause a mud flood or landslide. Okay, and then when there's no soil erosion, then the river will not become shallower due to the accumulation of the soil there. So this will prevent flash flood. Okay, if there's soil erosion, the soil will be brought down to the rivers, and then it will cause the river to be shallow, and then when the water during rainy weather, the water flows into the river, but when it's shallow, then it will tend to flood the place. All right, then so it prevents all these things caused by deforestation. It prevents global warming also, because we have lessened the increase in carbon dioxide concentration. Okay, so global warming can be lessened. Maintain forests as recreational areas, okay, because we didn't cut down the forest. So recreational areas are the areas for people to, enjoy the forest, walk in the park, and just relax. So reduce air pollution and water pollution, right? If we recycle the paper, because all this will not be thrown, the rubbish will not be thrown into the water, for example. And you can reduce air pollution because we will not burn the rubbish that contains the papers, right? Now, reduce the number of landfills because there's less paper as the rubbish. So every day, a lot of paper is thrown away in the plastic bags, right now. Whether it's paper used for writing or books or even tissue paper. So that increases the amount of rubbish thrown into the landfill, which is a place to collect the rubbish. Okay, so we can reduce the number of landfills when we reduce the paper that's thrown away as rubbish. And lastly, preserve the source of medicinal plants and food, which are the forests. Okay, the forest is the source of many types of plants that can be used as medicine by the natives also, and as food. Next is the recycling of used glass. All right, so if we recycle the glass again, then less new glass will be produced, need to be produced by the factories. More money and energy can be saved, okay, because to make glass, glass is made from natural or raw materials, that is sand, silica, silicon dioxide, soda ash, and limestone. And these have to be melted at very high temperatures to form the glass. So if we recycle the glass bottles and containers, then we will use less energy to melt the glass and form the new pieces of glass, or form the glass containers, to produce the glass containers. So it prevents depletion of minerals, and less fossil fuels will be used. That means because it will need to be burned, less fossil fuels will need to be burned to produce the glass. Now, what is the mineral used for making glass? It is silica or silicon dioxide, and you need soda ash and limestone. All right, so it will prevent depletion of minerals, and it will reduce air pollution because there's less burning of fossil fuels to manufacture the glass. It reduces water pollution because less glass products will be thrown away into the rivers. All right, it reduces the number of landfills, right, where the rubbish is thrown away, and it reduces exploitation of the earth's resources used to produce the glass, like all the sand and other substances used to make the glass. Now, by reusing plastic, what's the benefit of doing this? Less petroleum is needed to produce new plastic. Okay, so because plastic is made using petroleum, more petroleum in the earth can be preserved for future use. Less plastics will block the drains and cause flash floods. Okay, so a lot of plastic bags are thrown into the drains and rivers, and also into the sea. Now, because plastics do not decompose, they are non-biodegradable, so they can still be around after many years. The plastic bag, if you just bury it in the ground, okay, because it is not a natural substance that can be decomposed by bacteria or decomposers. So that's why we need to recycle the plastics. Less plastics to spoil the beauty of scenery, like on the beach. The plastic can be an eyesore. A lot of plastic bottles and plastic bags on the beach. It reduces air pollution as plastics release poisonous gas when burnt. Okay, so take note of that. And it reduces the number of landfills. Same point as for this one, G5. Okay, and S7, and just now P11. So altogether, even though you write 3 times, reduce number of landfills, you get only 1 mark. Okay, it reduces water and air pollution. Okay, because less plastic bags will be thrown into the rivers, less plastic bottles and plastic bags, and even plastic forks and spoons. Then it reduces number of animals that die when they mistake plastics for food. This is very true for the turtles in the sea. Whenever you see the plastic bag floating by, they thought it's a jellyfish. Then they eat it, and they may die from eating the plastics. Okay, because if the plastics in their stomach, they can't absorb their food, and they are not actually receiving food. Okay, when they eat the plastics, they are not getting food. So for if you mention air and water pollution for all the 3 parts of this question, you also awarded 1 mark only. All right. Okay, so total marks is 10 marks. So maybe 4 for the benefits of reusing paper, then glass another 4 marks, and then plastic can be maximum 4, but all add up must be, total marks can be only 10 marks. Okay, right, so that's all for this lesson. I hope that you have learned something useful for it. Do share, like, and subscribe. Thanks for watching, and goodbye.